Hello and welcome to another video in our course on genetics. In this video, we are going to talk about mutations. So what are you going to learn in this video? We are going to answer three important questions. What are mutations? What types of mutations do we have? And what causes mutations? We are going to talk about different types of mutations, such as somatic and germline mutations, insertions, deletions, missense, nonsense, silent mutations, and many more. After watching this video, you will also understand important terms like mutagen, deamination, or mutation rate. By the way, you can find written notes on what we're going to discuss today at medicinecanbeeasy.com. The link is also in the description. So what are mutations? A mutation is a change in the genetic information in an organism. For example, when a sequence of cytosine, guanine, adenine gets changed into adenine, guanine, adenine, we talk about mutation. Now, there are many ways by which mutations are classified. The first classification takes into account in which cells mutations occur. Somatic mutations can occur in any type of cell in the body except for germ cells. For that reason, they cannot be passed on to future generations. Germline mutations, on the other hand, occur in germ cells and do therefore pass on to future generations. The second classification distinguishes between gene mutations, which usually affect only one gene, and chromosome mutations, which change the number or the structure of chromosomes. Another classification considers the mechanism by which a mutation occurs. The simplest gene mutation is a base substitution, a mutation in which one nucleotide is replaced by another one. There are two types of base substitution: transition, when a purine is replaced by another purine or a pyrimidine is replaced by another pyrimidine; and transversion, when a purine is replaced by a pyrimidine or a pyrimidine is replaced by a purine. We also distinguish insertions, mutations in which one or more nucleotides are added, and deletions, mutations in which one or more nucleotides are removed. Insertions and deletions are dangerous mutations as they can lead to the so-called frameshift mutations. Frameshift mutations are changes in the reading frame of a gene. Simply said, these mutations change the way a gene is read. As you can see in the example below, a deletion of just one base, thymine, in the codon guanine-thymine cytosine will cause a frameshift mutation and different amino acids are going to be synthesized. The mutation can also change a sense codon, a codon that specifies an amino acid, into a stop codon and stop the protein synthesis altogether. There are also forward and reverse mutations. A forward mutation is a mutation that results in an altered phenotype. A phenotype refers to all the observable characteristics of an organism. A reverse mutation then reverts this altered phenotype into the original one. For example, a forward mutation could change the color of the eyes from green to brown. A reverse mutation would then revert the change from brown to green. Of course, in reality, this is much more complicated and this example is only hypothetical. Another classification considers the effect the mutation has on the structure of a protein. A missense mutation is a base substitution that changes the codon in a way that a different amino acid is synthesized. A nonsense mutation changes a codon that specifies an amino acid into a stop codon. And a silent mutation changes a codon that specifies a certain amino acid into a different codon that, however, specifies the same amino acid. A special type of mutation is the suppressor mutation, which suppresses the effect of another mutation. As you can see here, we have a base substitution that is going to result in the synthesis of threonine instead of serine. However, the suppressor mutation which changes guanine to cytosine is going to produce a codon that specifies serine again, and therefore will suppress the effect of the first mutation. Now there are two types of the suppressor mutation, intragenic and intergenic. The intragenic suppressor mutation occurs in the gene where the mutation it suppresses is located. The intergenic suppressor mutation, on the other hand, occurs in a different gene than the one where the mutation it suppresses is located. Another interesting type of mutation are the expanding nucleotide repeats. These mutations increase the number of copies of a set of nucleotides, usually 3 nucleotides. These mutations cause, for example, the Fragile X syndrome, in which the number of copies of the cytosine-guanine-guanine set of nucleotides located on the X chromosome increases up to 1,500 copies. Expanding nucleotide repeats are also the cause of Huntington disease, Kennedy disease, and many others. Now that we understand the different types of mutations, let's try to answer the question, what causes mutations? There are mutations that occur randomly, for example by an error in the DNA replication, and these are called spontaneous mutations. They can involve spontaneous chemical changes in DNA, such as depurination, the removal of a purine base from a nucleotide, depyrimidination, the removal of a pyrimidine base from a nucleotide, or deamination, the removal of an amino acid group from cytosine, usually producing uracil. However, mutations can also be caused by chemical, biological, or physical agents in the environment. These are called induced mutations. Some chemicals can induce deamination, which we've just mentioned. Then there are chemicals called base analogs that do have structures similar to the standard bases in DNA and as a result can be incorporated into newly synthesized DNA molecules during replication. What's more, chemicals called alkylating agents donate alk groups, for example methyl, to nucleotide bases, inducing all types of mutations. Chemicals referred to as intercalating agents, for example ethidium bromide, place themselves between adjacent bases in DNA and damage the helix structure, causing insertions and deletions. Causing insertions and deletions in replication that often result in severe frameshift mutations. Also, UV light can be quite damaging as it is absorbed by pyrimidine bases and it causes the formation of chemical bonds between adjacent pyrimidine molecules, creating pyrimidine dimers, usually thymine dimers, that often block replication. Finally, we should mention that the frequency of new mutations occurring in a gene or in the organism is called the mutation rate. And any agent in the environment that causes mutations is called a mutagen. Okay, so this is it. Thank you so much for watching. If you found this video helpful and you'd like to watch more videos on genetics or medicine in general, please subscribe. Thank you again and see you soon.